Hi guys, this is Ms. Romani, and in this lesson we will begin our exploration of the chemistry of life. Now we've reviewed some chemistry already, but chemistry is not alive. So in today's lesson, we will take a closer look at the field of biology called biochemistry. Biochemistry is, as the name implies, the chemistry of life. And biochemistry can be subdivided into three main areas of study. One deals with the chemical structure of molecules, which will be the focus of this first unit. Another deals with bioenergetics, or the study of how energy flows in organisms, and this will be the focus of the metabolic processes unit. And a third focus of biochemistry is about how DNA, as a molecule, can convey and pass on information in living systems. And this topic will be explored in our molecular genetics unit. Now, the discipline of biochemistry, this idea there is a chemical basis for life, is relatively new. Before the advent of biochemistry, a widely held belief was that of the principle of vitalism. Vitalism states that what happens in a living cell is unique to a living cell and can't be duplicated elsewhere. It was a false belief that living things have a unique life force that could create the products of life, one that could not be explained by chemistry. Now, this belief was proven false by Friedrich Wöhler. Wöhler was able to show, using ordinary chemistry, that he could make urea in a container in his lab. Urea is a compound made by living things. You are familiar with it because that's where the term urine comes from. Urea is found in urine. And because it was made by living things, it was believed to be made only by living things and only by cells and their unique vital force. Wöhler showed that the molecules of life can actually be made by chemistry. A few years later, the chemist Eduard Buchner isolated an extract from dead yeast cells, and then he used that extract to complete the process of fermentation, but he did it inside a flask, so he didn't use any cells. And in doing so, he concluded that biochemical processes do not necessarily require living cells, but instead are driven by some sort of special molecules that are found in cells. But if they're removed from the cells, the same process can be done outside the cell. And so because he discovered these molecules inside yeast cells, he named them enzymes. En meaning in, and zyme meaning yeast in Latin. So Buchner showed that fermentation, a process that was believed to be only possible using the vital force inside cells, could actually be performed outside the cell, essentially proving once and for all that there is nothing special or unique about cells and their molecules, well, at least not in terms of their chemistry. And this is huge, because once we knew that the chemistry of living things could be understood and could be replicated, we could begin to study it and use that information to better understand diseases and how to cure or how to treat them with chemicals, which is basically what the field of medicine is about. So biochemistry is all about studying the molecular composition of life. Take a human body, for example. It is made up of 60% water, which we already learned a little bit about, but the rest is mostly 4 macromolecules, and these are the ones that we will explore one at a time in the next few lessons. These are carbohydrates, fats or lipids, proteins, and nucleic acids. These are the biological molecules. They make up all living things on Earth and are necessary for their survival. They are the main source of energy. They are the means of storing that energy. They make up our body parts. And they are also the instructions that all organisms use to make those body parts and which are then passed on to future generations. When we look at the structure of these 4 molecules of life, we will notice that they all are carbon-based molecules. The skeletons of the biological molecules are made up of carbon atoms covalently bonded into large stable chains. Basically, life on Earth is built on carbon. But what does that mean? What makes carbon so special? What makes it the backbone of biochemistry? The answer to this can be partially found when we look at the electrons of carbon. Now, carbon has 6 electrons. The first 2 are in the inner shell, and the other 4 are the valence electrons in the outer shell. And as you may remember, these are the ones that are available for covalently bonding with other atoms. And since carbon, wants to have 8 electrons in its outermost shell in order to be stable, carbon atoms have a tendency to make 4 bonds. And these bonds can be single, or they can be double, or they can even be triple bonds. And the bonds can be formed with many different elements. And it's not just this ability to form many different types of bonds with different elements. Carbon is also a very small atom. So when it is bound to 4 other atoms, a carbon atom will then take on a very stable 3-dimensional shape. So it is this ability of bonding with a variety of other atoms, including itself, and to form a very stable shape that makes carbon so special. Another thing is that carbon-to-carbon bonds can form very stable chains and rings, which make them excellent building blocks for very large molecules. So any molecule whose basic shape is made up of carbon and hydrogen is called an organic molecule. And since life on Earth is made up of these organic molecules, we call it carbon-based. But is it possible that there's life that is not carbon-based? Well, if it is, we certainly have not found any. But let's explore that possibility for a minute. Is carbon the only element capable of building the large, stable, and complicated molecules of life? Probably not. Silicon, for example, which is right under carbon in the periodic table of elements, shares many of the same chemical properties as carbon. Like carbon, it has 4 valence electrons and is able to form 4 very stable covalent bonds. But a lot of the bonds formed by silicon are actually weaker than those formed by carbon, and would probably not be able to make the stable, large, complex molecules that carbon is able to make. Still, because carbon and silicon have so much in common, some scientists think that it could theoretically be possible that life on other planets could be silicon-based. As a matter of fact, this possibility was explored in an episode of Star Trek back in the 1960s. So, in this episode, right, Kirk and Spock beam down to a planet called Janus 4, following a distress call, and discovered that more than 50 mine workers had been slain by a hideous monster. It turns out that this monster was actually a silicon-based life form called a Horta. And Horta feed on rocks and secrete some sort of acid that could destroy human flesh. Now, then Spock got to mind meld with the Horta and discovered that the Horta was actually a mother, and she was simply trying to protect her eggs, which the minders had been destroying because they thought they were rocks. Anyway, we got sidetracked here, but back to carbon. So carbon is the backbone of biochemistry, right? Well, the carbon chains that form the backbone of organic molecules are called hydrocarbons. They are kind of a skeleton of these molecules and are made mostly of carbons bonded to other carbons and bonded to hydrogen. And attached to these hydrocarbon skeletons, we will find what we call functional groups. And these are small chemical groups like this OH right here. The hydrocarbon skeleton gives the biological molecules their general 3-dimensional shape, but it is actually the functional groups that participate in the chemical reactions and make the biological molecules unique and special. There are 6 functional groups that are of particular importance in biological molecules. You will learn about these and a few others in grade 12 chemistry, but for this course, we will only focus on these 6 because they are found in the macromolecules of life. For example, if you look at a molecule of a sugar called glucose, you will notice all these OHs that are attached to the carbons. This is a functional group called hydroxyl, and this functional group is the reason why sugar dissolves in a cup of coffee. Think back on our lesson on polar bonds. The oxygen hydroxyl will have a partial negative charge, and the hydrogen will have a partial positive charge. That polarity is what makes any molecular region where a hydroxyl group is found hydrophilic in nature. And that's why sugars are water soluble and can dissolve in your morning coffee. Now, in organic chemistry, any molecule that contains a hydroxyl group is often called an alcohol. And you may already know that alcohols can dissolve in water. This structure right here is called ethanol. And ethanol is an alcohol molecule that is found in wine, in beer, in pretty much any alcoholic beverage you might be familiar with. And basically is the only alcohol that is safe for human consumption. Or I guess relatively safe, since alcohol is not completely safe for human consumption. In a few moments, we're going to explore another alcohol that is completely unsafe and very, very toxic, And you will see how the functional groups kind of have a role to play in that toxicity. Now, speaking of coffee, as we were a few moments ago, here's a very important molecule to some of us, caffeine. Caffeine has many functional groups, but we will focus on this one right here. This is called a carbonyl group, and it is a carbon double bonded to an oxygen. Like hydroxyl, it is also a polar functional group and makes molecules that contain it hydrophilic. Carbonyl groups come in two forms depending on where it is in the molecule. If the carbonyl group is at the end of a hydrocarbon chain, it is called an aldehyde, and the carbon is bonded to a hydrogen as well as the oxygen. If, on the other hand, the carbonyl group is found in the middle of the molecule or in the middle of the ring, then it is called a ketone. Now, this molecule here is one of the building blocks of fats, and it is called a fatty acid. It is acidic because it has a functional group called carboxyl that is acidic in nature. As a matter of fact, carboxyl groups are often called carboxylic acids, and they're basically a carbonyl group with the carbon bonded to a hydroxide group. Now, when dissolved in water, this hydrogen right here tends to break off, adding protons to solution and making the carboxyl group a weak acid. In its ionized form, when the carboxyl group has dissociated and released its hydrogen ion, the carboxyl group is then negatively charged. And like I mentioned before, carboxyl groups can be found in fatty acids like this one, but they are also part of a structure of amino acids. Actually, many organic acids are weak acids because they have a carboxyl group. For example, this molecule right here in the example is called acetic acid, and it's basically the acid found in vinegar. Okay, so far we've looked at three functional groups: the hydroxyl group, the carbonyl group, and the carboxyl group. I want to now take a moment to maybe illustrate the biological effect of changing the functional groups in a hydrocarbon, how it changes the chemical properties of the molecule, and as a result, the effect it can have, say, on the human body. So let's start with a molecule that may be familiar: ethanol. Ethanol is an alcohol that has two hydrocarbons and a hydroxide group. Now, like I mentioned earlier, ethanol is the alcohol found in beer and in wine and vodka and pretty much any alcoholic beverage that people consume. And as you may know, alcohol gets people drunk. What it does is it directly affects brain chemistry by altering levels of neurotransmitters, which are the chemical messengers that transmit signals between nerve cells. And this leads to the common immediate symptoms of intoxication, like impulsive behavior or slurred speech or poor memory or slowed reflexes. Now, despite any stories you might have heard about drinking coffee or taking a cold shower, you cannot sober up by doing any of these things. Once alcohol is in the bloodstream, it can only be eliminated by an enzyme in our liver called alcohol dehydrogenase. Now, this enzyme metabolizes ethanol into acetaldehyde by replacing the hydroxyl group with a carbonyl group. Now, you may be thinking that this is a good thing because alcohol is obviously not great and it impairs the brain and people need to sober up eventually. But acetaldehyde is actually quite toxic. It damages body tissues, it can impair memory, it can cause a fast heartbeat and headache and an upset stomach, which are basically the symptoms of a hangover. It is also a carcinogen, which means that it can cause cancers. So of course our bodies will not want to keep this molecule around. Thankfully, we have another enzyme, aldehyde dehydrogenase, that very quickly replaces the carbonyl group with a carboxyl group, turning acetaldehyde into a non-toxic acetic acid. Now, here's something really interesting. It turns out that more than a third of East Asian people, when they drink, they experience something called facial flushing. They obviously get really red in the face and they feel ill when drinking too much alcohol. It turns out that this is due to an inherited deficiency in aldehyde dehydrogenase, which then fails to convert acetaldehyde into acetic acid. And now, depending on whether or not the person has inherited one or two copies of the faulty gene, they will have varying degrees of severity of the condition. Regardless, the toxic effects of acetaldehyde are more pronounced in people with the faulty gene, which makes them more prone to alcohol-related cancers. On the other hand, though, people with the gene tend to drink a lot less because, well, alcohol makes them feel awful, so oftentimes it's just not worth it. Now, here's another interesting thing about alcohol metabolism. Remember how I said that ethanol is the only alcohol molecule that is relatively safe for human consumption? Well, here's an example of a very dangerous alcohol molecule: methanol. Dangerous amounts of methanol can sometimes be found in bootleg or homemade alcohol, so you should avoid those definitely. The structural difference between ethanol and methanol is actually tiny. Just this single carbon is the difference in their structure. But the differences in their effects on the body are actually huge. Methanol is poisonous to the human body. And not poisonous in the way that ethanol is poisonous, like you can get alcohol poisoning if you drink too much alcohol. Methanol is poisonous because even small amounts of it can kill you. And if it doesn't kill you, it can cause permanent blindness. But why is that? Well, it turns out that it's not really about the difference between ethanol and methanol, but the differences in the molecules that these alcohols are metabolized into. Turns out that the same enzyme that breaks down ethanol into acetaldehyde will convert methanol into a molecule called formaldehyde. And this process is actually somewhat slow, which explains why the symptoms of methanol poisoning can sometimes take hours or days to manifest, because it takes time to turn methanol into formaldehyde. And it's not the formaldehyde that is actually extremely dangerous and toxic, although formaldehyde is not great for the body. This is not the part that kills you. But formaldehyde, once present, will very quickly be turned into formic acid by aldehyde dehydrogenase. And it is this formic acid that is the toxic part of methanol poisoning. Formic acid is the chemical found in ant stings. So if you ever get stung by one of those like fire ants, it is formic acid that is in that compound. A buildup of formic acid essentially damages nerves in our eyes called the ocular nerves, causing the blindness that is often associated with methanol poisoning, and can very often lead to death, even in small amounts in the body. Okay, so on that note, let's take a look at some more functional groups. So here's an amino acid. As we saw earlier, it has a carboxyl group, but amino acids also have an amino group, which is the one functional group so far that does not just contain carbons, oxygens, and hydrogens, but it actually contains nitrogen. And it actually consists of a nitrogen attached to two hydrogens. An amino acid is both an acid and a base, but it's the amino group that makes it basic. So amino group is a weak base. In water, the amino group will pick up a proton, an extra hydrogen from solution, which will then increase the pH of the solution, and the amino group will essentially become charged in the process. Now, this is the amino acid cysteine right here. And I'm not putting this one here to show carboxyl groups or amino groups again, although in this particular diagram you can see that both the carboxyl group and the amino group are found in their ionized forms. But I'm actually showing you this amino acid because it is the only amino acid that has a sulfur atom in it, in its structure. And this sulfur atom forms a functional group called sulfhydryl, which consists of a sulfur bonded to a hydrogen. Now, sulfhydryl groups are important in determining the shapes of proteins because when two sulfhydryl groups come close together, they can actually covalently bond and form what is called a disulfide bridge. And a disulfide bridge can help stabilize the three-dimensional shape of a protein. So they're actually very important for protein shape, and we're going to revisit this functional group when we take a look at proteins in more detail. Finally, this is a nucleotide. You may recall from grade 11 biology that nucleotides are the building blocks of DNA. Nucleotides contain a phosphate group. A phosphate group is a negatively charged ion that consists of one phosphorus atom surrounded by 4 oxygen atoms. And it is easy to recognize because it is the only functional group that contains phosphorus in them. Phosphate groups make molecules acidic. They are incredibly important since they make up part of the backbone of the structure of DNA and the structure of RNA. And there are 3 of them in the energy molecule called ATP. So phosphate groups are also an important part of the structure of phospholipids. These are the main molecules that make up the cell membrane. So that's the 6 functional groups that you will find in the 4 molecules of life. And now let's see if you can recognize them. Pause the video now and check to see if you can recognize the hydroxyl groups, the carbonyl groups, both the aldehydes and the ketones, the carboxyl groups, amino groups of hydro or phosphate groups. I'll give you the answers once you're done, but for now, just pause it, maybe draw out the structures, circle and label any of the functional groups you can remember, and we'll see how you do. Okay, so here are the answers. In this first structure, you will find a carboxyl group, an amino group, and a sulfhydryl group. Now, some of you maybe went a little bit above what was asked, and you maybe named this as an amino acid, which would be correct, and more specifically is the amino acid cysteine. Now, the second structure has a carbonyl group, more specifically an aldehyde, and 2 hydroxyl groups. We're going to become more familiar with structures like this later on, and this is actually a triose sugar, a sugar that is made out of 3 carbons. This next structure is also a sugar. This one has a carbonyl group called a ketone, as well as a whole bunch of hydroxyl groups. And then last, this is a nucleotide, and the nucleotide will have a phosphate group and a hydroxyl group and an amino group. Okay, so hopefully you did well and you were able to recognize some of these functional groups. When we explore biochemistry in the next section, you will see how these functional groups play a role in the structure of the macromolecules of life, the 4 main macromolecules of life, which are carbohydrates, lipids, proteins, and nucleic acids, as I mentioned earlier in this lesson. And one of the first things that we will discover actually about these macromolecules is how they are built or put together. Most biological molecules are very large and are built by assembling smaller molecules called monomers into these larger molecules, which we actually call polymers. Polymers are basically these large molecules that are made up of many monomers, often in a repeating pattern, and they are put together by a process called polymerization. And the process of polymerization happens via a type of chemical reaction called dehydration synthesis, although some texts would actually call them condensation reactions. And the name of the reaction gives us a couple of clues. Let's look first at the dehydration part of the name. When you dehydrate something, you are removing its water. So this tells us that the reaction involves the removal of water. And synthesis means to build or to put together. So a dehydration synthesis reaction is one that removes water to form bonds between molecules in order to build a larger molecule from the smaller molecules that make it up. Like all chemical reactions in the body, the reaction is actually helped or catalyzed by an enzyme. And here's an example of a dehydration synthesis reaction we will explore in more detail in the lesson on proteins. This one will join 2 amino acids, the monomers of proteins, together by removing water. And remember that the functional groups of the molecules are the ones that are involved in the chemical reaction. So let's watch what happens when the carboxyl group of one amino acid reacts with the amino group of another amino acid. The enzyme removes the hydroxyl group from the carboxyl group and one hydrogen from the amino group to form water, and then forms the bond. Now, the opposite reaction is also extremely important in living things. So as much as we can build large molecules to, say, build new cells and body parts, we also need to be able to break apart larger molecules, like, for example, during digestion. So to do this, we use the chemical reaction called hydrolysis, which means adding water to break something, in this case, to break a bond. So hydro refers to the addition of water, or hydrating the bond, and lysis is a word in biology that means breakdown. So during hydrolysis, an enzyme will add water to the bonds in the polymer and break them, releasing the monomers. And again, we can see the process of hydrolysis being used here to break the bond between two amino acids. Water is added, the carboxyl and the amino groups are reformed, and the bond is broken. So to recap, dehydration synthesis reactions build polymers by removing water, and hydrolysis reactions break them by adding water. And these reactions are responsible for the formation and breakdown of all the macromolecules we will explore in the next few lessons. Oh, and one more thing before we end this lesson. In chemistry, you may have learned that there are many ways that molecules can be represented. However, since biology mostly involves larger molecules like, say, glucose, for example, we will actually just focus on the structural formula. And there's one thing that you need to know about drawing structural formulas in biochemistry, as well as in organic chemistry. And that is that biologists and organic chemists are lazy when it comes to drawing out large structural formulas. Well, maybe saying that they're lazy is a bit unfair. Let's just say they're efficient when it comes to drawing out large structural formulas. So, for example, when they draw large molecules like this glucose, say, or this fatty acid, they will sometimes do something like this right here. So what's happened in this structure here, this first molecule at the top is glucose, this next one is also glucose. But notice that the carbon atoms have been omitted from the structural formula at the bottom. And that is because it is kind of understood in organic chemistry and in biochemistry that the carbon atom is the backbone of the molecule. So any points in the structure is basically an implied carbon. And some representations take it even a step further and will also imply not just the presence of the carbons, but also the presence of any hydrogens that are bonded to those carbons. So that a skeletal representation of a fatty acid ends up looking like this. Or the skeletal version of the glucose ends up looking like this, with just the hydroxyl groups and that extra carbon at the top, but any carbons are omitted in the ring, and any hydrogens attached to those carbons are not included in the structural formula. Or let's take, for example, a look at an amino acid. Here's an amino acid drawn with every atom present, and here's what it would look like when we stop drawing the carbons or any associated hydrogens that are not part of a functional group. So notice that the functional groups are still there. The functional groups need to be included because they're not the hydrocarbon skeleton of the molecule. They are the ones that are involved in the chemical reactions, and they're going to give that particular molecule their unique properties. So that's it for today's